Hello everyone, welcome back. In today's lecture, we are going to start a new topic, electrospray ionization, commonly written as ESI. ESI is a very important ionization technique used in mass spectrometry. It is known as a soft ionization technique, which means it can convert molecules into ions without breaking them into too many fragments. In simple words, electrospray ionization takes a liquid sample and converts it into gas phase ions using a high-voltage spray. This technique is especially useful for compounds that are large, polar, or sensitive, such as biomolecules, peptides, proteins, and pharmaceutical compounds. Now, let's understand the basic idea. In ESI, the sample is usually dissolved in a suitable solvent. This liquid solution is passed through a small capillary or needle. A high voltage is applied to the needle, which creates a fine spray of charged droplets. These charged droplets move toward the mass spectrometer. As the solvent evaporates, the droplets become smaller and smaller. Finally, gas phase ions are produced from the analyte molecules. These ions then enter the mass analyzer for detection. One major advantage of ESI is that it works directly with solution phase samples. That means it can be easily combined with liquid chromatography, also called LCMS. This is why ESI is widely used in modern analytical chemistry, pharmaceutical analysis, biochemistry, and proteomics. Another important feature of ESI is that it can produce multiple charged ions. For large biomolecules, this is very helpful, because when a molecule carries multiple charges, its mass-to-charge ratio, or m/z value, becomes lower and easier for the mass spectrometer to measure. So, even large molecules like proteins can be analyzed effectively using ESI. Let's quickly review the main points from this slide. First, ESI is a gentle process. It preserves molecules with less fragmentation. Second, it can produce multiple charges, which helps in analyzing large molecules. Third, it works in the solution phase, so liquid samples can be used directly. And fourth, it is LCMS ready, meaning it can be easily coupled with liquid chromatography. So, the key idea is this. Electrospray ionization converts liquid samples into gas phase ions using high voltage while keeping the molecules mostly intact. Now let's discuss the principle and mechanism of electrospray ionization, or ESI. The basic principle of ESI is very simple. It converts a liquid phase analyte into gas phase ions using high voltage, droplet evaporation, and ion release. In ESI, the sample is not first converted into a solid or gas manually. Instead, the sample solution is directly introduced into the ionization source. This is one reason why ESI is very useful in LCMS analysis. Now, let's understand the mechanism step by step. Step number 1 is sample introduction. The liquid analyte enters through a very fine metal capillary. The sample is usually dissolved in a suitable solvent, and it flows slowly through this capillary. Step number 2 is high voltage application. A high voltage is applied to the capillary. This creates a strong electric field at the tip of the capillary. Because of this electric field, the liquid forms a fine charged spray. This spray contains very small charged droplets. Step number 3 is charged droplet formation. These charged droplets move toward the counter electrode. Inside each droplet, analyte molecules are present, along with solvent molecules and charges. At this stage, the sample is still mainly in droplet form, not fully converted into gas phase ions. Step number 4 is desolvation. Desolvation means removal of solvent. As the charged droplets travel, the solvent starts evaporating. Because of evaporation, the droplets become smaller and smaller. As the droplets shrink, the charge density on their surface increases. In simple words, the same amount of charge is now present on a smaller droplet, so the repulsion between charges becomes stronger. Step number 5 is ion release. Finally, gas phase ions are released from the droplets. These ions enter the mass analyzer, where they are separated according to their mass-to-charge ratio, written as m/z. So, the full sequence is: liquid sample enter through the capillary. High voltage creates a charge spray. Charged droplets move forward. Solvent evaporates. And finally, gas phase ions are released and detected. The key concept is that ESI converts solution phase analytes into gas phase ions with little fragmentation. This is why ESI is called a soft ionization technique. It is especially useful for large biomolecules, polar compounds, peptides, proteins, and LCMS-based analysis. Now let's discuss the instrumentation of the electrospray ionization source, also called the ESI source. The ESI source is the part of the mass spectrometer where a liquid sample is converted into gas phase ions. Its main job is to create a fine charged spray, remove solvent from the droplets, and send the final ions toward the mass analyzer. In this slide, we can see the main parts of an ESI source. Let's understand them one by one. First is the capillary. The capillary is a very fine metal tube through which the liquid sample enters the ESI source. The sample is usually dissolved in a suitable solvent and introduced slowly through this capillary. So, the capillary acts as the entry point for the liquid analyte. Second is the voltage supply. A high voltage is applied to the capillary. This high voltage creates a strong electric field at the tip of the capillary. Because of this electric field, the liquid coming out of the capillary changes into a fine spray of charged droplets. This is the most important step in electrospray ionization because the high voltage helps convert the liquid sample into charged droplets. Third is the nebulizer. The nebulizer helps in spray formation. It assists the liquid sample in breaking into very small droplets. In simple words, the nebulizer supports the formation of a fine and stable spray, so the ionization process becomes more efficient. Fourth is the drying gas. The drying gas helps evaporate the solvent from the charged droplets. As the solvent evaporates, the droplets become smaller. When the droplets shrink, the charge density increases, and finally ions are released from the droplets. So, drying gas is very important for desolvation, which means removal of solvent. Fifth is the ion optics. After ions are formed, they must be guided properly toward the next part of the instrument. Ion optics focus the ions and direct them in the correct path. This improves ion transmission and helps produce a clear mass spectrum. Sixth is the analyzer inlet. The analyzer inlet receives the ions from the ESI source and sends them forward into the mass analyzer. Inside the mass analyzer, ions are separated according to their mass-to-charge ratio, written as m/z. Now, let's understand the sequence shown in the diagram. The liquid sample enters from the capillary. High voltage creates a charged spray. The nebulizer helps form fine droplets. Drying gas evaporates the solvent. Ion optics focus the ions. And finally, the ions enter the analyzer inlet for mass analysis. The legend at the bottom helps us understand the particles. Green represents droplets, purple represents charged ions, gray represents neutral analyte, and blue represents product ions. So, the main idea is that the ESI source gently converts a liquid sample into gas phase ions using voltage, spray formation, drying gas, and ion focusing. Now, let's discuss the features, advantages, and applications of electrospray ionization, or ESI. ESI became very important in modern LCMS because it can directly analyze liquid samples and gently convert them into gas phase ions. First, let's look at the key features of ESI. The first feature is that ESI is a soft ionization technique. This means it causes less fragmentation and helps preserve the original structure of the molecule. The second feature is that ESI produces multiply charged ions. This is very useful for large biomolecules, such as proteins and peptides, because multiple charges reduce the mass-to-charge ratio, or m/z, and make large molecules easier to detect. The third feature is that ESI works with liquid samples. The analyte is dissolved in a solvent and introduced directly into the ESI source. Another important feature is that ESI is highly compatible with LC-MS, which means liquid chromatography can be connected directly with mass spectrometry. This makes ESI very useful for separating and analyzing complex mixtures. ESI is also suitable for polar compounds because polar molecules can easily form ions in solution. Now, let's move to the advantages of ESI. One major advantage is that ESI is very good for analyzing large biomolecules. Compounds like proteins, peptides, nucleotides, and other biological molecules can be studied effectively. Second, ESI provides high sensitivity for trace analysis. This means it can detect very small amounts of compounds in a sample. Third, ESI causes lower fragmentation, so the molecular ion information remains clearer. Fourth, ESI is useful for complex mixtures. When combined with liquid chromatography, different components of a mixture can be separated first and then detected by mass spectrometry. Fifth, ESI helps in molecular weight studies, especially for large and sensitive molecules. Now let's discuss the applications of ESI. ESI is widely used in proteomics and peptide analysis. It helps scientists study proteins, peptides, and their structures. It is also used in metabolomics, where small molecules and metabolites in biological samples are analyzed. Another major application is pharmaceutical analysis. ESI is used to study drug molecules, impurities, degradation products, and drug metabolites. ESI is also useful for environmental samples, such as detecting pollutants, pesticides, and contaminants in water or soil. Finally, ESI is used in clinical and forensic testing, where biological samples need sensitive and accurate analysis. So, the key point of this slide is ESI is especially valuable when the sample is polar, fragile, or too large for hard ionization methods. Finally, let's summarize the Electrospray Ionization ESI lecture and highlight the key takeaways. ESI is a soft ionization technique that converts liquid samples into gas phase ions while preserving the molecular structure. Here's the complete sequence in simple steps. Step 1. Liquid sample. The analyte enters the ESI source through a fine capillary. This is the solution phase starting point. Step 2. High voltage. A high voltage is applied to the capillary. This creates charged droplets from the liquid solution. Step 3. Desolvation. The solvent evaporates from the droplets, shrinking them and increasing charge density. Step 4. Ion release. Gas phase ions are released from the droplets. These ions are now ready for detection. Step 5. Mass analysis. The released ions enter the mass analyzer, where they are measured according to their mass-to-charge ratio, m/z. Main takeaway: ESI gently transfers molecules from solution into the gas phase. This makes it ideal for LC-MS analysis and for studying biomolecules like proteins, peptides, and metabolites. In simple words, ESI keeps fragile molecules measurable without breaking them into too many fragments. This is why ESI is widely used in modern LC-MS systems, proteomics, metabolomics, and pharmaceutical analysis.